Shalom, and thanks for the invitation. I will not repeat what the people said of how lovely and great and vivid it is to be in such a meeting. I'll start by saying that I will do it shorter than planned, promising, because there's coffee and break, and I know that we need it. And that my talk is going to be divided into two pieces. One is the motivation and approach that we are currently using within IBM, and it will give you a wider context to the type of research we are doing. And then I'll show specific results that were performed by marvelous teams. Some of them are here. So while I'm starting, maybe Chen, Yara, Michal, can you raise your hands? Are you in the room? I see Yara over there. Michal and Chen may be outside. They told me they might need to listen from there. Thanks. The new world of after COVID, with COVID, it's not really clear what, is a world where AI is no longer sufficient. It's no longer sufficient because typically when we say the typical AI is machine learning and prediction algorithms, so I mean, there are many different ways to define AI, but if I limit it to this type of machine learning algorithms that learn from the past and predict the future, then it might not be sufficient because the world is shifting so quickly, and we've seen that in the industry, how the industry had to adapt and adjust based on the major shifts in the world. Working from home that we all experienced, supply chain problems, and many other things. So when we looked at IBM and what does it mean to the new world, to the industry and to the world, we sort of went back to the Sir Francis Bacon, who introduced the scientific method 400 years ago, and talked about a cycle of decisions, and talked about the fact that when you apply the scientific method, which is what we did the whole day today, there's a question you try to answer, and a study that is performed in order to answer that question, and then comes up an hypothesis, and then a testing of the hypothesis, an experiment, an assessment of the results in order to conclude that we have made a real important discovery. And I think that Uri Shalit, Eran Segal, the rest of the professors who talked today illustrated that very well. They talked about their study, the question they wanted to answer, and without repeating their talk, when I listened to it, it was very easy to pinpoint where was the answer to the question that comes up with an hypothesis. And in fact, causal inference is a key technology that is being used in order to generate hypotheses, where you try not only to find something that you are able to predict, you try to identify causal relations and to be able to pinpoint that there's something that seems to cause a result. And if that something that seems to cause a result is a real, there's a real causation connection there, then there's an hypothesis that we can test with an experiment. And that's the only way to validate the hypothesis. I'm going to talk about drug repurposing, and the motivation for repurposing drugs is clear. If we think about the expense of how much it takes to develop a new drug, is according to some estimates, more than $1 billion, close to $2 billion, and vast majority of it happens at the very beginning, in the preclinical and the first phase. 50% is the second and third phase, and of the funds, of the investment, so it's an order of $1 billion. And if we could minimize or eliminate this need of establishing safety and finding the connections through preclinical studies, we might be able to save a lot of money and reuse existing drugs. So that's the motivation, that's the question we asked ourselves. Can we identify existing drugs that might benefit specific outcomes associated with diseases? We focused, and I'm already almost switching to the second part, we focused on Parkinson's disease. And the question that we asked ourselves together with a team from Teva company, we asked the question of, given that so many people are suffering from Parkinson's disease, can we identify drugs that these people take because of comorbidities, because they have other conditions, and find out that this drug, the drug for the other condition, is beneficial for the Parkinson's disease? So the question was very clear, and then we launched a study, and we ended up with an hypothesis We didn't end up with what we would like to do, which is an experiment, but this is still an ongoing study, and there's a vast work on this topic within IBM, our IBM research team. And again, I'll ask Ishai to raise his hand. He's leading the team, and he's here, and Rom, who joins us lately, is with him now. So, how do we address the question of finding new indications for existing drug? And I will illustrate everything. The running example would be Parkinson's disease. We designed the question in such a way. We looked back at vast data sets, so millions of people that we have access to. And in this data that we looked back, this is data coming from US, data that EMR's data of Explorers or market scan data of claims data, and I'll show in the next slide the names very clearly. We looked at this data of patients, and we wanted to design the study in such a way that we divide the group into those who use the drug and those who are not using the drug. So now remember, we are looking in this example on Parkinson's disease patients. They take drugs for their Parkinson condition, but God forbid, these people often also suffer from other conditions, for instance, sleeping problems. So they take other drugs. And we went through all these other drugs that appear in our data, and we asked ourselves, for these other drugs, let's look at all the people who use this other drug compared to people who did not use that specific other drug, but did have similar conditions. So they used drugs for the same condition, but not that specific that we are going to test. And we created this user, non-user cohorts, and we analyzed that. We have a tool, and you can see here the reference to the technology that was created to develop this tool. And we apply this tool on the first step, which is mapping the data into some structured information, set of features. So the data is typically, and that goes back to, I think, what Professor Rishalit said about can you identify a clear point in time at which the intervention happened? In our case, yes. This is the index date, the date at which we see in our data that the person started to take a drug. Then there's the question, how much do we know about these patients? Of course, we don't know everything, and we still had to make some assumptions. But we do have a lot of information in the EMRs of Explorers. We have a lot of information about their medical records and their diagnoses and tests and so on. And of course, these other drugs that they obtained. And in the market scan data, which is claims data, we have somewhat similar data, wider data set, but less informative per person. So we had these two different data sets from the US. We created a tool. This tool is open source, and everybody is welcome to come and access that. And it's recorded that the story of what includes in this tool is available on archive right now. And this tool supports then the causal analysis with validation tests on the balancing of the data. So there are many different methods to apply the causal analysis. And we selected a very specific method that I'll show in a minute. We first mapped the data, as I said, and we applied this causal analysis tool, and I'll show exactly in what way we did it in this specific data. So the method is laid out in this Jamia paper, and the specifics of the data and of the technology. So I'll start with the data. We had close to 100,000 Parkinson's disease patients in our data. That's massive data. Of course, at the end, when we look at the more than 200 different drugs that these people take not for their Parkinson's condition, they are not such big groups. The cohort is smaller. So we had some rule of thumb that says that if the people who are using the data are less than 100 patients, we're not going to analyze the data. It's too small. It's not likely to generate significant results. So we had these limitations on the data that we started with. And we analyze this data and just automatically, and it's part of the application of the scientific method in the industry in many different areas. As I said, Netflix are using us for experimenting with all of their products. Inditex, which is the owner of Zara and Pull & Bear, are using experimentation in the way they sell their products, their dresses, and other clothing. So the experimentation is very common now in the industry. And what we did here, we wanted to make sure that we have significant results. So we wanted to make some condition and constraints on the size of the data that we are looking at. And we went back into the data and we analyzed it with a specific method of causal inference that I lay out here. There's the procedure that you can see, but may I say that we used, in principle, we use Kaplan-Meier algorithms, and we asked ourselves, because the data is censored, not all the patients are definitely in the data from start to end. They might have moved from one database to another. So the data is censored, and we wanted to analyze the difference between the outcome of the trial drug to the outcome of the alternative drug by reweighting with inverse propensity scoring and comparing the outcomes through empirical results that are using these weights. So you can see in the first equation up there, the probability for trial drug outcome for people who take the trial drug to have a certain outcome compared to the alternative. And by the way, the outcome that we looked at several outcomes. The outcome that we focused at the end was Parkinson's dementia. So the question is, are there drugs not yet known that might affect Parkinson's dementia? And we estimated, we wanted to calculate the probability, which is not something that is available to us, so we made estimates that are time-dependent for the outcome, for the data, after weighting the data with these weights that you see in the second equation. The results are that we found two drugs that seems to be very effective or somewhat effective, I don't know if to say, somewhat significantly affecting on the Parkinson's dementia. One is rosagiline, that is known for Parkinson's motor problems, and it was found in our data, both Exploris and MarketScan, so you have validation here from two different data sets, was found to be effective. And similarly, zolpidem, which is a drug for sleeping problems, was found to be effective for Parkinson's dementia. What we found after, so this was automatically done with more than 200 drugs. And the whole idea, again, in the industry of the cycle of discovery that is the ability to do it at scale and at speed. So a key element here is that we had a scalable system that is based on these components that I mentioned before that enabled running tests on more than 200 drugs and performing that on these 100,000 patients of Parkinson's in Exploris and in MarketScan. In one, it was a little bit less. In the other one, it was a little bit more. And from all of these automatic analyses, we did use knowledge base. The knowledge base we used was to organize the drugs by their mechanism of action and the way they're supposed to be effective. So we did group the drugs, and when we compared zolpidem, it was comparing the usage of zolpidem to usage of drugs from the same group according to the definitions, the RX norm definition of groups. So you can see here that it's the names of a group, the NO4 and the NO5 appearing on the graphs, and you can see that the blue line and the red line shows a clear deviation of the effectiveness between those who took the user drug, the drug in trial, and those who used the alternative drug in all 4 graphs, which are 2 drugs in 2 different databases. This was automatically derived. Then we looked at the literature, and we were somewhat surprised, but not very surprised to see that there's a paper that talks about the value of zolpidem for Parkinson's dementia based on a case study of 1 person, and an ask and an urge to test it in a much larger data set. So that was not a way for us to focus on zolpidem. That was retrospectively studied. So you can see here a summary of the study in a graphic picture, and the paper that was published a few months ago in the Frontiers of Pharmacology. Thank you very much. I left you 3 minutes. Thanks.